Simon, thank you very much. Thanks everybody for being here. I know that coming to seminars can be an arduous thing. I'm glad they provide you free food. It's the most important thing you'll take away from this hour is your stomachs being full. So it's great to be here to talk. I'm going to preface my talk by saying that I changed the title a little bit from how a fly sees the world to how we see how a fly sees the world. And given the fact that I'm here on Georgia Tech's campus, some of the technological and more sort of engineering aspects of my work that I have to belabor when I give talks to molecular neuroscientists, I'm going to speed through very quickly. But if you have a question, go ahead and raise your hand and interrupt me. I'm happy to not get through the whole seminar, is what I'm trying to say. So I'm interested in flies. I'm interested in insect vision. I have been since I was a kid and an undergraduate where I worked on dragonflies, and as a grad student I worked on hawk moths. And I'm interested in insect vision because of what you see here in this picture. So this is an extremely coarse camera. This is a 25 by 25 pixel camera. You would never buy a cell phone that had this imaging system, yet this animal can use this imaging system to see in almost 360 degrees of arc. It can't see through its own body, but it can see everywhere else, and it can see faster than your visual system by at least an order of magnitude. So these animals actually have really high performance vision, and I find that quite interesting. In neuroscience, there's a few broad questions that can generally be applied to my research. People want to know, how does visual attention work? How do you choose to attend to me rather than the sandwich? How does the visual system discriminate visual features from the surroundings? How do you identify that butterfly that's skimpering across the landscape against the foliage? And how is visual perception modified by context? You probably have some intuitive sense that your sensory experience depends somewhat on the context within which you're embedded. The smell of pizza has a very distinct and attractive aroma when you're hungry, but after you've just eaten a lot of it, it itself can repulse you, right? So context makes is very important in our understanding how the brain processes sensory information. In flies, can we apply some questions that might begin to address some of these broad themes? So in my lab and in talk today, I'll talk a little bit about the fact that flies, they're these miniature high-performance sensory motor processors. All right, you wouldn't want to buy the cell phone that does this, but man, wouldn't you like to have a robot that does this? How is visual gaze stabilized during flight? This is one of the questions we'll look at. What are the algorithms and functional mechanisms of visual feature detection? And by the way, this is going to apply to mammalian systems as well. Almost everything I talk about in fly vision can be applied with analogs to mammalian retinal visual feature detection. And finally, how is visual feature detection modified by other senses? I'm not going to get there. There's no way I'm going to get through all these slides, but offline we can talk about that. The methods that are required for my research, I thought this would be important to demonstrate because most of you have a more technical background and how does my lab integrate these things? And the methods that are required come from lots of different fields. We have people in my lab that were EE majors and they build instruments and electronics to build virtual reality flight simulators for flies. We have BME and physicists that come in and do systems identification and control theory to derive formal algorithms with which we can use to build hypotheses to test underlying neural circuits, for which we use molecular cellular developmental biological approaches in molecular genetics, fluorescence microscopy, immunohistochemistry, in vivo optical imaging. We build transgenic flies to detect, read, and write to neurons. We don't do all of this quite as elegantly as we would like, but we're getting there. And finally, we have ecologists in the lab who think about what is it that these animals, where have they evolved? What have they evolved to do? Why is their visual performance so spectacular? It's because they've evolved over millennia to do certain behavioral things in natural ecological contexts. And we have to keep that in mind. In fact, as a sensory neurobiologist interested in essentially how insect eyes work and maybe how insect noses work, in my career, I realized that you cannot uncouple those questions from the organismal behavior. Why is that? So it turns out, of course, that you've got some sensors, and they process visual and olfactory input. You may be able to formalize these inputs with control theoretic framework. These sensors provide premotor processing to centralized networks that produce a rhythmic motor code that activates musculoskeletal mechanics and wing kinematics to build aerodynamics. And that's what propels the animal through space. And that's important because it's the animal's movement through space that generates the sensory input in the first place. So as a sensory biologist, I'm trapped by all the mechanics and all the theory and how the plant works and how the control systems work and how the feedback works on this operation, which was initially where I started my career. So it's inextricably linked, this whole process. Now, of course, most of you in this room would say, of course, that's obvious, but let me tell you, like neuroscientists don't get this, right? Not all of us get this. And it's because it's very hard to try to incorporate these kinds of integrative approaches into a single research program. Okay, so flies. So what do flies do? Well, flies fly. That's the coolest thing they do. That's not what they prefer to do, by the way. They'd rather sit and groom. Flies would much rather lick themselves than fly around, okay? So if I was really a sensory ecologist, I'd study licking behavior, but I find that to be a little problematic and boring. I like flight behavior. So in this system, you see, this is an animation of real data. The dot is a single Drosophila flying around in a 1-meter tub, and it's being tracked by a pair of video cameras. And what you see is this stereotypic pattern of flight behavior. This is played back at real time, where you see the animals, they fly straight, and these straight paths are interspersed by rapid rotations of the body, rapid reorientations of the body. And we call those turns saccades for the functional analogy to our own gaze-stabilizing eye movements. Flies can't move their eyes inside their head. They instead produce saccades by rotating their whole body. Now, what are the sensory cues, visual cues in particular, and the motor commands that modulate saccadic flight behavior? Was a question that we recently undertook. Well, it turns out that the animals are saccading toward features of their environment. So if this is sort of a natural environment that a fly might grow up in, it's composed of these vertical edges. Their flight behavior is essentially cursorial mid-air. They fly these flat trajectories, and they saccade in the yaw dimension. So you can see that when we provide just a really simplified forest in this arena of 3 stripes, the animal's trajectories tend to map paths in between those objects. They fly toward one. It approaches on the retina. They don't want to crash into it. They turn. They orient directly toward another one. They fly across the arena toward that one. It expands on the retina. They don't want to crash. They turn, et cetera. So they tend to be heading toward these 3 vertical objects in the arena if you unwrap the arena and look at the probability of where they're heading. So they head toward these salient vertical features of the environment. These are landscape features. These are perches, right? These are things that are naturally occurring in all of their natural world. And it turns out that vertical edges are really important to all visual systems. So a really nice paper that came out of a vertebrate, a primate visual processing lab in 2011 demonstrated that by scanning millions of photographs of natural landscapes and actually finding the edges in these photographs, and then looking at the probability of the orientation of those edges, you get a bimodal distribution where there's many more vertical edges in natural landscapes than any other orientation. And it turns out if you were to map the population of orientation selectivity neurons in your own cortex, it follows exactly that same bimodal distribution. We've evolved in landscapes that are driven by gravity. Things grow vertically, things land vertically. Your visual system is really well tuned to horizontal motion of vertical edges. So flies too. So how do we study saccadic flight behavior in flies in a more controlled environment? We stole from human visual psychophysics. So here on the right, you're looking at a fly glued to a pin, suspended in a magnetic field, and surrounded by an LED display. So the fly is fixed in place, and we can video track it from below, but you can see that the animal can't fly away, yet produces these sort of saccadic reorientations of its body. Okay, and this was directly stolen from human psychophysics literature in the 50s, where similar kinds of arenas were built for human observers and their eyes were video tracked. Now I'm going to show you a video of a very simple experiment. We wanted to look at how saccadic tracking was influenced by these landscape features that flies naturally orient toward. Okay, and so we built a very simple experiment where the LED display that surrounds the fly produces this vertical striped pattern that's sort of random, broadband, spatially broadband, roughly naturalistic, and then one patch of that bit of the arena, the vertical stripes, just rotate around against the stationary background. And the flies are going to react to the rotation of this visual figure. And you'll see the data here. Here you'll see this is the image of the fly from below. This is the head and the abdomen, so the animal's facing this direction. You'll see the angle of the object in blue and the angle of the fly in white, and the error between the two over time in this graph. You can actually see the wings, or maybe it's kind of washed out on your display. Basically, everything you need to know about figure tracking in flies is represented by this movie. And what you see is the staircase of saccadic turns in white, where the animals rapidly reorient and then stay still, rapidly reorient and stay still. perfectly canonical saccadic pursuit. And the error distribution is overall very consistently low. So you can see very clearly this saccadic pursuit of visual objects in this sort of quasi-naturalistic setting. So when we analyze this a little bit deeper, what we show is what, in this figure, you see exactly what you just saw in the movie. Oh, the angle of the bar is continuously changing smoothly. The animals are following it in time with this blue staircase of saccades, with this little sort of sawtooth of error, angular error between the animal's frontal midline and the position of the bar. And they produce these rapid modulations in angular velocity, these saccades, in order to chase and track this visual object. And what's intriguing is that in between saccades, so this little segment of time in between these rapid angular rotations, the animal stays basically still. Okay, the bar is rotating continuously, but the animal is not rotating with it, right? So it stays still between the saccades, which is very much like what you saw in the free flight movies, okay? And that's going to be an important feature of the whole control system that'll come up again later. So flies, they track objects with saccadic orientations of their body. That actually is exactly how humans track objects with saccades of their eyes. This should not be surprising to any of the ecologists or visual ecologists in the room, because the same kind of saccadic behavior is seen in goldfish and rock crabs, and basically any animal that has an image-forming eye that tracks things in the world uses a saccadic strategy. The evolution of why that strategy works is a fascinating topic of discussion. It's a signal processing, signal noise kind of a topic of conversation that we could have offline. But it's a very clearly converged mode of very efficient visual tracking. Now, what causes, what triggers the saccades? What about the angular distribution of where the object is triggers the saccades? And if you just look at the count of saccades as a function of the error angle, in other words, how far away from midline, or where was the object located, this vertical bar located when the saccade was triggered, and you see that basically at about 45 degrees off midline is the sweet spot. That's where, when the bar sweeps through about 45 degrees off midline, the animal triggers a saccade, either to the right or to the left, depending on the direction of the object. Now, what happens when you rotate the ground smoothly instead? So here's a regime in which you don't see saccades at all. If the entire panorama is smoothly rotating, then the fly produces a smooth pursuit strategy. You don't see the saccades as regularly. You see this very smoothly controlled angular rotation of the body that follows the retinal slip generated by this striped panorama going around. And you can see that in these incredibly smooth trajectories that are just perfectly aligned, yet somewhat delayed from the rotation of this visual pattern. Occasionally, you see some saccades that serve to generally catch up with the pattern as the display moves too fast. The animal's gain is slightly lower than unity, and the animal will produce a catch-up saccade. This is precisely the same result. If I put you in one of these flight arenas, and I've actually done this, I put my head right in these things, and they really kind of make you sick, but because your vestibular system is disconnected, and your eyes involuntarily track the whole panorama, and occasionally nystagmus saccades are performed to sort of catch up or reset. So here we have this fundamental dichotomy in flies. They are using smooth optomotor, or optokinetic would be the analog in primates and humans, movements to stabilize the retinal image. You do the same thing. And you're using these rapid saccadic reorientations to track salient objects against that background. Now here's a bit of data that comes out of the optomotor experiment. You just saw this trajectory. You saw that as the background is rotating smoothly, the animals essentially track with saccades that are interspersed with this very smooth overall tracking effort. If you rotate the display at different speeds, you see something really interesting. So in the top, middle, and bottom, what we're showing is the retinal slip on the retina. Okay, so we're moving the pattern at 135 degrees per second, or 90 or 45, and then we're plotting how much slip across the retina in the fly's frame is being produced. How much motion does the animal's visual system actually detect during these experiments? And what's really intriguing is that you basically see that the offset, either positive or negative, depending on the direction of the stimulus, is really similar for all three of these. It's about 20 degrees a second. Okay, so the closed loop feedback system is using the optic flow independent of the velocity of the pattern in order to essentially maintain equilibrium. And at a very low retinal slip speed. It turns out that most of the neuroscience, most of the visual neurobiology done in fly visual systems, they present velocities 10 times higher than that to a recording of a fly or to a behavioral test of flies, and they test the velocity domains that are an order of magnitude larger than this. Yet the fly's inherent control loop reduces all of that and maintains a very effective, rather uniform, very low velocity regime on the motion detecting system in the brain. Now what happens when you combine these two systems together? You've got smooth tracking going in one direction and saccadic tracking going in the other direction, right? So we're rotating the panorama smoothly, counterclockwise, while we revolve the figure smoothly clockwise. And what you see is what looks like a superposition of both control efforts. So in this graph, the figure is moving along the blue line. The background is moving along the white line. And what you see is most of the time the animals are tracking the figure with these little saccadic steps. Sometimes, occasionally, like here, the animals actually track the background rather than the figure. But they don't do both. They do one or the other. They don't do both. So the gaze stabilization in the wide field domain is not corrupted by figure tracking. The systems are hybrid and switched. Okay? One is operating or the other is operating, but not both. So Jean put together, and this is published work, so I'm not going to go into a lot of detail. He put together a control theoretic model where you collapse the dimensions, of course, of the animal, and it's got some body dynamics and a reasonable neural controller, and these various elements of feedback and feed-forward interactions. And he basically built two different controllers, one for the velocity of the wide field panorama, and one for the position of the small field visual object. And both of them involve a smooth tracking component, or sorry, the wide field one involves a smooth tracking component, and a saccadic component, although this is less well understood, whereas the small field sensitive one has no smooth pursuit component. It's all driven by the execution of torque spikes out of the motor system that actually produces these saccadic body rotations. And basically coding up this model and testing it in rather simple ways, moving a bar at 3 different velocities. I didn't go into the parameters with which the animal actually is stabilizing. Suffice it to say that as the error angle changes, it's the integrated error, the spatiotemporal integral of these waveforms that produce a threshold command to trigger saccades. So the data show that over integrated error, as the velocity of the figure changes, the error changes with the velocity. Higher speed generates more error, but the integrated error is constant. So the animals maintain a constant integrated error of the visual figure, and the model shows basically the same thing. And they do that by modulating the strength of the torque in proportion to the speed of the object. Okay? So the higher the error, the higher the torque, in order to correct for and maintain constant the spatiotemporal integrated error of the object off of midline. And look, the takeaway from this is really just that it's a hybrid controller, and that a hybrid controller really captures the overall behavioral phenomena well, and it's extremely testable. Okay, it's very testable. I'm not going to show you any of it. I'm not going to test it at all for you today. Watch the career of Jean-Michel Mongeau, who just started his own lab in mechanical engineering at Penn State. So what I'm going to shift and talk about how do flies see these things? How does the fly visual system? It's this crappy 25 pixel camera, 25 square pixel camera. How does it see these really complex kinds of visual motion? And let me demonstrate what that looks like. So this is the same movie you just saw. But on the bottom panel, I've unwrapped the visual display, the cylinder that the animal's steering around. And I've blown it out so that you can watch what the retinal projection looks like from the fly's perspective. This is perspective corrected flow that the fly sees. And what you see is that that bar, that discontinuity generated by that bar motion, which you can see, right? Your eyes find the object, right? If the movie stops, the object disappears because it's only defined by its motion. Now it's gone, right? There's no contrast cue. There's no luminance cue. There's only a relative motion cue. Sorry about that. I want to keep that going. So you see that basically when the object, it kind of stays over here by about 90 degrees. It's because when it hits about 45 degrees, the fly saccades toward it, right? So it's always displaced from midline. That's number one important feature. Number two is that while the animal's tracking that cue, it's experiencing massive amounts of optic flow in the retina. The whole visual world is constantly moving while the animal's tracking that bar. Occasionally, the animal will switch from tracking the bar to tracking the background. When the background stays still, that means the animal's following it. So occasionally you see signatures of the trajectory I showed you. The animal's either tracking the bar or it's tracking the ground. But let me tell you something. No model of motion vision in flies can see that object in this scene. The elementary motion detector that's been used since 1950 is a beautiful biophysical model for motion vision. An array of those local motion detectors integrated over space can't do this task. How does the fly see this? So flies produce, like humans and many other animals, they produce saccadic eye movements to track objects. They produce smooth motor movements to track the panorama. They will track objects against a moving panorama using a hybrid controller that splits the inputs of those two visual pathways. And that the dominant model, and I'm not showing you all the theory, but the models for motion vision in flies are terrible at extracting this really complicated visual stimulus. How do flies see this thing? We developed a bunch of experiments to test some extremely challenging visual stimuli that were inspired by primate vision, and we adopted them to fly vision against my own judgment, because I thought if we build these stimuli and the motion detector can't see them, and we show that flies can see them, I'm going to get so much flack from the rest of the vision community. And let me tell you, it was brutal. It was really brutal. But these are the experiments we came up with based on the ethology, based on what we know flies can perceive. So here's this sort of unwrapped image, and then in space-time, here's a space-time graph. And this will illustrate why the motion detectors don't see it. So here's a randomly flickering background. And here is a bar that the one you just saw. It's made up of a patch of the background. When it slides back and forth, see this line in space-time? That means that there's a dt by dx correlation. There's a direct correlation in space and time. That's what motion detectors can see. This bar, you have no trouble tracking, yet a motion detector cannot track this, at least the correlation-based motion detectors, because there's no space-time correlation. Here's a bar where the texture moves one way, the bar moves the other way. So all the space-time correlations go in the direction opposite the bar. So the motion cues are moving opposite the figure cues, okay? A model based on, so a correlation-based motion detector will see that this bar is moving left, and this bar is moving to the right, and this one is moving to the left, which is exactly opposite what the stimulus shows and what your brain trivially recognizes. How do we figure out how, and I'll tell you the punchline is flies see all of this. I've never built a visual stimulus that I can see that the flies can't see. Never once. I can build plenty of things that the flies can see that I can't, but that's because they see 10 times faster. So when I put my head in there, all I see is flashing, blinking. It's making me nauseous. I'm determined that I coded the stimulus wrong. I go back to the MATLAB code, and I'm like, I make a movie, go real slow. No, it's right. And I put my head in the arena. I can't see it. And my postdoc's like, it's totally wrong, boss. It's totally wrong. It can't be right. And I'm like, just run the flies. And the flies give me the data that I knew was going to be in there. Anyway, how do we detect how these animals see what they see? Well, we have to go to a more constrained behavioral system. So here we've got a fly glued to a pin and suspended now under an optical beam, which casts a shadow of the wings onto an analyzer that basically produces a voltage. There's just two photodiodes with a mask that conditions the output so that the voltage coming out of each of the two diodes is proportional to the amplitude of each wing. So if you subtract the left from the right overall voltage coming out of the sensors, you have a signal that's exactly proportional to yaw torque. Okay, you can use that time-varying signal to couple to the feedback to a feedback controller for the actual display. And here's this LED display, and it looks really coarse to you, but to flies, this is like HD TV. And there's a single bar, and the animals are actively steering the bar. So there you can see the wings modulating a little bit. Every bit of motion that you see, here's just a pattern of bars. And same idea, the animal has closed loop control over the bars. Here you can really see the wing kinematics modulating rapidly, kind of unstably, as it controls this visual display. All of the movement of the of the display that you saw was controlled directly by the fly. So, the idea here is we know precisely where and when on the retina visual stimuli are operating. We know exactly what the animal's reaction is. Jean's going to take the magnetic tether system into this domain like tomorrow, and then I'll have to stop giving this talk. But for now, I'm good. We did a systems analysis in the following way. So, okay, you're all familiar with this, right? You put white noise into some system, you don't know anything about it, you get an output. You can cross-correlate these two signals, and you get some transfer function, in this case in the time domain, right? So what we did is we made a bar out of two different stimuli. So on one sequence, we're not going to use real white noise, we use an M sequence, which is a maximum length shift register sequence, which produces discrete little pulses of the display to the left and right. So that the display pops right 1 pixel and left along this time course. So here's our F, our input, and we have two different sequences. One of them moves the texture inside the bar, which you saw in the movie, okay? So it's jittering. The other sequence moves the bar, okay, independently. So we have two inputs, a single compound output, but we cross-correlate the output, which is the output of the wing beat analyzer. So the animal is steering left and right, in response to both this compound set of stimuli. And we can derive, right, the impulse response for the EM, or elementary motion inside the bar, and the FM, the figure motion of the window itself. And you do this for a bar that's centered near midline, and you get these two kernels. And this has basically units of change in the wing beat amplitude per impulse seconds, or if you cancel the units, it's basically volts per degree, but voltage is such a stupid term because this is an uncalibrated output. The point is, ignore the units. It's not that meaningful, but the time course is. So we're deriving these two impulse responses, and we do this in the following way. Here's a video that shows how it works. The fly is looking at a stimulus that looks like that over time, and it's moving its wings, trying to stabilize both of these two signals simultaneously. And then we do this over space. We put the bar on the left. We run both M sequences. We get the outputs. We do the cross-correlation. We measure kernels. Then we put measure the kernels. And then we pseudo-color them and make time go down. And then we fit them. We smooth them out a little bit. Move the bar to here. Repeat. Move the bar here. Repeat. And we finish with what we call spatiotemporal action fields, which I named. My students think I'm ridiculous. I'm like, you know what that is, boss? You're giving us a staph infection. And I'm like, so what? I don't care. I'm using it. It's like spatiotemporal receptive fields, big thing in neuroscience. We have the spatiotemporal action field of the fly. And it's really cool. It shows the dynamics, both in time and space, in response to motion within that little bar and motion of the bar itself. And I love the fact that the dynamics couldn't be more different. Couldn't be more different. In one case, you have a very rapid response that decays rapidly over a few, 100 milliseconds, and is sort of tuned in the frontal field of view. The response to the figure itself, independent of the motion contained within it, has this real slow DC onset. I mean, really slow. This is 2 seconds. And it actually inverts. The sign inverts in the periphery. And what's really cool is that the maximum difference between the peak and the trough is right here at 45 degrees, which I think is not coincidental compared to where in the magnetic tether, where does the animal trigger a saccade? Where is the figure when the animal triggers a saccade? It's right there in the sweet spot of this FM staff. So I was always really excited about this work. I thought this is going to revolutionize the field. One guy in the field told me, this is a game changer. This is a classic paper. And he immediately cited it, and he started to use the technique a little bit. And then freaking crickets, man, like nobody in my field. I give talks, and this molecular biologists in the audience are like, M sequence. What's an M sequence? It was really hard to get any traction. But I think it's about to come full circle. And the reason I like it so much is that it was incredibly predictive, right? This is what you do systems analysis for, is to be able to predict how the animal is going to behaviorally respond. So when you superimpose these two staffs, and you can make a prediction, so you can measure the animal's steering response to this oscillating bar, which is in black. And you get an envelope of standard deviation in gray, and then you superimpose the prediction whereby you basically just convolve this in space and time with the staff, and you get this output. Like, it's trivial. And it's really, really well predictive, right? There's an R value of 0.97. Like, I didn't think that was possible. And then if you use a theta bar, now this is the weird stimulus where, right, the bar moves right, the texture moves left, and there's the animal's steering response. They definitely see it, and the staff prediction follows beautifully. And then we did an experiment where we just used M sequences to drive these two stimuli and outside of the staff derivation. So if you just randomly move these two around, you can beautifully predict the fly's response to both. So the animal's superimposing this FM system and this EM system to track objects, okay? And what happens, though, when you do this kind of very simple experiment? You know, this is an undergrad that did this. Well, what if you make the bar small? She read a paper that said, small objects repel flies. What if we make the bar small, you know, and measure staffs? And I thought, oh, boy, this is going to be a little tricky, but let's try it. We did it. I'm not going to show you the staffs, but one thing that came out of it was that while we were doing these experiments, we were recording little torque spikes. Now, in the staff analysis, this is like linear. We don't want these nonlinearities, so we ignore the saccades that might be produced measuring, you know, these two kernels. But we have them stored. And so if you look at where the saccades are produced in space. So in this graph on the top, what you're looking at is the torque spike amplitude when the object is on the left visual hemisphere or the right visual hemisphere. And then, on the top is torque to the left, bottom is torque to the right. And when the object is in the right, they torque to the right. When the object's on the left, they torque to the left. Okay, great. So this is exactly what you saw in the magnetic tether system. Behavioral people wouldn't have believed this as strongly as they believe the magnetic tether system, but it works. When you shrink the bar, look what happens. The distribution of saccades flips. So that now, when the object is on the left, they torque to the right. When the object is on the right, they torque to the left. So just by changing the vertical extent of this object, we completely flip the sign and gain, if you will, of the output of this motor center that drives tracking behavior. Why would this happen? You know, I don't really know. The ethologist in me says, all right, well, vertical edges are really strong. They're in the environment. You fly around. You want to go perch. You want to shoot gaps. You're navigating through these vertical features. But when you compress that thing into a small two-dimensional object, that's danger. That's some kind of approaching threat. It's a fly swatter, it's a bird, most likely it's a dragonfly, and so that becomes an aversive thing. So it's a really simple algorithm. Elongated edges, track it. Little short edges, steer away from it. Now, okay. In the magnetic tether, just to follow this up really quickly, right? So again, the tall bar, you know, they track after it with some delay. But when you shrink it to a small object, basically it repels them. They track ahead of it. So the small object produces larger amplitude saccades. They're different dynamically. They're distributed in a different direction. It's a completely different motor output, right? Essentially. So the behavior classifies this object and this object completely separately. Okay? That's an important point. Behaviorally, the animal classifies this thing and this thing completely distinctly. All right? It's important for the underlying neurobiology. Okay, let me just check on my time here. We're doing good. We have till 12, right? I want to leave time for discussion and why do you do this and how long have you been doing this and the crazy questions. How do you glue flies to a stick? I mean, I can anticipate them all. So let's get into the neurobiology a little bit, this is actually pretty cool. So the fly brain, this is work that's been studied since the time of Cajal. So fly neural architecture has been examined for over a century. So on the right, these are Golgi impregnations of the optic lobe of a fly. And what you see is, and this color is not real, obviously it's not in the brain, but there's a lamina, the retina is out here. So each of these little streaks is a single facet of the compound eye. Then there's the lamina and a medulla and a lobula and a lobule plate. So there's 4 optic ganglia. And they process information in a retinotopic manner. That means that, and you can see that here, one little cartridge of the facet of the eye sends its axons here into this little cartridge in the lamina. They send axons here into the medulla and so on. And then you get these retinotopic projections onto this rotated structure called the lobula plate, where the red neuron might integrate the input from a whole bunch of these things and detect, for example, horizontal movement of the whole world. Okay? The primate visual cortex has pyramidal cells that do basically the same thing. So a lot of work has been focused on the motion detecting pathway here in the lobula plate. And that's why what I've indicated here is that, so this is a Drosophila brain rotated in 90 degrees from this view and stained for a marker for a synaptic protein. And you can see the layered structure of the medulla, the columnar structure of the medulla, the layered structure of the lobular plate. This region here has been very clearly identified as the EM center, the elementary motion detecting center in the brain. This is where the neurons are that detect the movement of the entire visual panorama. This has been known since 1965, okay? The mechanism by which these computations occur are just now being formalized. So we know a lot about wide field motion vision. We know a lot less about object detection. In fact, basically what I'm going to tell you today is everything the fly world knows about object detection. And it occurs in this formally enigmatic structure called the lobula, which is extremely complex. So we go after this question of how do these various circuits process motion in a very simple way. We replay the dynamics that we record behaviorally into a physiological preparation. In this case, we're using a 2-photon excitation imaging microscope. I could talk about that offline. It's very cool. In this case, it's a quiescent preparation. The fly is glued to a stage. A little window is cut open in the back of the head, and it's in a water immersion bath. And we have the animal suspended in front of one of these LED displays, exactly the same thing that we use to drive the behavior. Again, to us, it looks really coarse. To the fly, it's HD TV. And we use some genetic tools to express a calcium indicator that fluoresces in the presence of calcium when the neuron depolarizes. And in this case, in a single neuron in a region of the brain that has 100,000 cells. So we can record the membrane, the proxy of membrane potential in a single cell, or even just a neurite of a single cell in response to these kinds of visual stimuli that you've already seen. So here's the canonical wide field detecting neuron. You rotate the display, and the cell fluoresces, and it turns out that the time course of that fluorescence means that it's integrating membrane potential, which is kind of cool. That stimulus pops membrane potential up to a static level that's maintained the entire trajectory, but the calcium transient increases along a perfect time course of integration. That's really important for the synaptic communication of what that cell does to control behavior, but I'll talk about that separately. These cells have been really well studied. They're in the lobular plate. We know a lot about what they do. So switching to this formerly enigmatic region of the brain called the lobula. Okay, so the lobula has these really weird neurons called lobular columnar cells. They're columnar, which is normal. The fly brain is, it's a compound eye. So each facet has a column of information that comes in and is processed in each layer of the optic lobe. And then the neuron that I showed you in the previous slide has a huge dendritic arbor. It samples a whole bunch of those columnar inputs, and it says, the world is turning right, and it fires off a big calcium signal when the world goes right. And when the world goes left, it's inhibited. But its output is integrating all these inputs, right? Here, these neurons, and what I'm showing you on this slide is a composite of fluorescently labeled the whole population of lobular columnar cells. And what you see very clearly is a whole bunch of color stacked on top of each other in the lobula, but then in the central brain, all the yellow cells converge to this one region. All the purple magenta cells converge to this spot, right? All the purple cells converge here, and then they cross over to the other side of the brain. All these secondary yellow cells converge to this little spot. There's 50 to 60 to 100 to 700 individual columnar neurons in each color. But all the outputs of any one color converge into one fist, one glomerulus. Glomerulus is from the Latin root glomus, which means ball of yarn. And in the olfactory system, it determines exactly, your olfactory system is built on the principle that all the receptors for your aunt's perfume land in one glomerular ball of yarn in the brain. And when that thing lights up, your brain behind the glomeruli know that that's your Aunt Denise. Okay, that's how you detect her, all right? If they're all lit up together, you avoid her, right? But if only a few of them are lit up, it smells great. I walk into Denise's house at Christmas, and I can smell her perfume. And she comes down the stairs, and she gives me a hug, and I'm like, right? But so the glomerular architecture of multiple receptors converging onto one glomerular input is really well known in the olfactory system, and it's really bizarre in the visual system because all these inputs here in cyan are retinotopic. There's one neuron here, another here, another here, another here, another here. They're all sampling from a small region of space one small column, yet their outputs converge here into this ball of yarn, and all the retinotopy is lost. So, like, what does that mean? That's a profound question I can't address completely, but we're working on it. Suffice it to say, in my mind, I saw this as being a way to encode feature space in a way that might be functionally analogous to the way the olfactory system encodes odor space. When this glomerulus lights up, that's one odor. When this glomerulus lights up, it's a second odor. When these, in combination of a third, that's a third odor. The dimensions of the odor space, right, might be number of glomeruli factorial or more, right? This is how I think of the glomerular organization of these lobular columnar cells. So let's take a look at a few of them. We screened a whole bunch of them for the kinds of visual stimuli that I've been presenting. So here's LC11. This is beautiful architecture. These are individual columnar neurons, and they all converge onto this foot of a glomerulus. LC12 neurons all converge onto their glomerulus. LC15, sort of similar geometry, exact same locations in the lobula, different glomerular outputs. And when you screen them for these kinds of visual signals, a tall bar, a little short bar, or the panorama itself, this is what you get. You get kind of a sparse activation profile. LC11 responds minimally to an elongated edge, but goes crazy for a small object. Totally zero response to the thing that drives the lobular plate crazy. LC12 responds to both of these stimuli similarly, and LC15 seems to filter edges of bars rather than small objects. None of the other 17 LCs that we screened see any of this at all, right? So this is really specific and high fidelity, very sparse representation of these really important visual features. Why is this so cool? Okay, so Hubel and Wiesel won a Nobel Prize for recording from the feline cortex. And they identified cells that had extremely precise response profiles to objects. And the most fascinating one was the so-called hypercomplex cell. And this is the method they used to identify a hypercomplex cell. They've got a cat anesthetized. They've got an electrode in the brain. They're recording single units extracellularly. And the animal's watching a screen, or in this case, is looking at a piece of paper on a wall when the experimenter has a flashlight. Don't you wish you could do these experiments now? So here, I'm just going to play the video, and there's sound. So I've identified the spatial receptive field. This is an audio amplifier of the spikes, obviously, right? Elongate the edge? Nothing. Really lovely. Canonical hypercomplex cell. The hypercomplex receptive field is defined by the fact that it's essentially size-tuned, right? Where you get an end-stopped inhibition generated by an elongated edge. You get the maximum response for a very small object, but as you elongate the edge, the response drops off. And look, I mean, these guys, they celebrated. They won the Nobel Prize for this work, right? It's totally fantastic, right? So jump forward. Here we go. Fry Lab, circa 2016. We have our little fly. This is our prep. It's all controlled. We spent a lot of time and energy. I wish we could just, use a little spot and put it on the screen, and then so it doesn't quite work that way. But we're imaging now from LC11 dendrites. And in the lower panel, you'll see the response as the bar crosses the receptive field drawn in red. So the bar response is essentially undetectable at the level of the dendrites. And when you shrink that bar down to a small object, yeah, that's the response I like. That's it. That's what I'm talking about. That's what I did. See? And I'm like, this is really cool. So then you graph this out, and you show the response increases as the object is really small, but as it gets larger, you get an active inhibition. And we did this, and then this is our version of celebrating. Mehmet Kelish was the grad student in the lab who did this work, and this is a party at my house afterward. Yeah, I didn't expect the Nobel Prize or any such kinds of things, but to me, like the cortical field is really complicated. It's hard to work on these circuits. It's hard to work with these animals. It's hard to develop reagents where you can isolate mechanism. But in flies, we have the techniques. So here's an LC11 neuron. Okay, here's what it's made of in a cartoon fashion. So there's individual cells that sample from lots of different retinotopic inputs, up to 90 we've calculated. Okay, so but the thing is, we want to measure, for example, you want to know how much dendritic overlap is there. So if there's only 70 neurons, there's 700 columns. Each one has to overlap and sample from a huge columnar input array. Do the input arrays overlap, and does that have a functional consequence? Well, how do you figure that out? Because the genetic reagent that labels these guys labels all of them. But we can cleverly trick through molecular chicanery. I'll steal this term from Richard Axel. We can build flies that have a heat shock construct that basically stochastically flips all the green labeling to red in single cells. Okay, because we don't want to label just one cell. We want to label all the cells in green, but we want to label one or two of the cells in red so we can see how much of the dendritic overlap there really is. And so you can actually do this measurement directly. You can measure exactly how much overlap there is. And does any one columnar neuron have its own visual field? And the answer is no. I won't go into the details, but the answer is no. I was hopeful the answer was yes, but science is about failure, right? I mean, this is what we do. Research, if we're not failing, we're a factory, right? Where it's all about, so we failed. But we're figuring this out. We're trying to figure out the functional implications of the columnar overlap. What about the layered structure? How does this help us understand feature detection? Rotate the lobule on its side, it's layered. We know that it's layered. Each of these LC neurons innervates specific layers. It's exactly how the visual cortex works. Where the processing of various information takes place is layer specific. We stained we basically made a genetic region where we could label LC11 in green, and this is an antibody for acetylcholine. You get this really strong labeling of acetylcholine directly at the edge of the dendrites of LC11. So they seem to receive huge amounts of excitatory input. VGAT labels for the vesicular GABA transporter. So this is a transporter molecule for the inhibitory neurotransmitter GABA, again overlaid with LC11 in green. And you can see that basically the LC11 dendrites behind the dashed line heavily label with this vesicular transporter for this inhibitory neurotransmitter GABA. So that gives us some ideas about how the excitation comes in and inhibition sculpts the receptive field properties. More directly, we can do this really cool thing, and I'm almost done, so I'm not going to take much more of your time. So here we've generated a fly where we're using so GAD1 is the gene that encodes GABA in the entire brain. We're coupling the gene that synthesizes GABA to a GCAMP, which is this fluorescent marker for calcium activity, but it's tagged with SYNT, which is a protein localized only in synaptic terminals. So we're putting the GCAMP, tethering it to the synaptic terminals. So we're only going to report the presynaptic outputs of any cell in the brain that is GABAergic, okay, that produces GABA. And then we're just going to display on our LED screen a whole bunch of different visual stimuli. And this is more for context than science, right? But look at all these flashing flashes. This is the medulla. This is the lobula. This is the lobula plate. And you can see layer-specific flashing. You can see waves of retinotopic spread in response to some stimuli. The reproduction on the video is not ideal here, but you're looking at basically there's 100,000 axons that cross over this image, and if you did a pan-neuronal recording with GCAMP in all the neurons, you'd never see any of this patterning. But we can actually measure the active calcium responses in the neurons that are producing the inhibition in space and time everywhere in the visual system at once, which led us to identify this cool experiment where we're looking at now the patterns of inhibition in response to the stimuli that would otherwise activate LC11. This is the entire lobula that's labeled here. Okay, now what are the patterns of inhibition in the lobula in response to a small object? That's the inhibitory, those are the inhibitory release sites, if you will. In response to a bar, much stronger. In response to the wide field grading, huge, right? So GABA signaling is definitely sculpting the size selectivity in this feature detecting neuron. And you can analyze the layer-specific effects of this by looking at a mean fluorescence image in response to the object, the bar, superimposing them, and noting that there's layer-specific activation of the small object compared to the bar and the grading. And this leads us toward a better understanding of the molecular logic that drives feature selectivity. And the final note that I will make and then finish is just to say that these experiments led us to identify a neuron in red which innervates only in the lobula. It doesn't send projections out. It doesn't get any inputs in. It's entirely intrinsic to the lobula, and it totally surrounds the LC11 dendrites. And it itself, so the neuron is now labeled, the cell bodies are in green, and we're looking at an antibody for the GABA synthesizing gene, and they completely overlap, right? So this neuron in red that's surrounding the LC11 is inhibitory. It's GABAergic. It's releasing GABA. And if you record the activity of this thing, you get this really cool effect. In black is the response to a small object in LC11. And in red is the response to the small object by this local interneuron, right? 0 response. To a bar, LC11 shows a weak response. The 29G10 shows a relatively strong response. And to the wide field motion, right, the red responses are very large, whereas LC11 is very small. So it looks like one cell has the perfectly complementary receptive field properties of the other. And to test whether that's actually has a molecular logic, we can put in a PTX. So this is a pharmacologic agent that blocks GABA A receptors. And when you do that in the blue traces, you basically recapitulate the red traces in the LC11 neuron, which is not yet causal, but it's extremely robust evidence that the inhibitory signaling required for feature detection by this neuron is being driven by this counterpart, this local interneuron. And this is a level of resolution that doesn't exist in most other visual systems yet. So we're very excited about this, and it gives us a really clear understanding of where in the brain these really sophisticated and complex visual features are encoded. I didn't get to talk about it, but there are LC neurons that are very sensitive to these theta bars and the drift balance bars that I showed before. This graduate student, when he came, he's like, I'm not going to do the white noise. I don't want to do it. I want a black bar, and I want a little black box. And if you don't like that, you can get somebody else to work on this project. He's like, I don't want to work with these complex stimuli. I don't understand systems identification theory. I'm a molecular biologist. I can do the imaging, but please don't make me do any white noise, analysis. And I was like, fine, Mehmet, use a black bar, use a little black dot, knock yourself out. So there's something for everybody, right? There's something for everybody. So I've told you a whole lot about the behavioral algorithms by which flies are using to track objects or avoid them. I told you a lot about systems identification measures that demonstrate that there's 2 separate subsystems, one for the motion generated by these objects and one by some other higher order statistical feature of the objects that doesn't stimulate the motion pathway. I told you that the lobula plate does not encode these stimuli, but that they are encoded by these very strange population of neurons in the lobula, these columnar cells that form these optic glomeruli. And we hope soon to kind of close the feedback loop, and I'll be able to tell you that if we sort of optogenetically activate these cells, there's a whole little segment of the talk, 10 more minutes, that does some optogenetics, but I don't have time. But we're coming toward the point where we could actually read and write to these cells, provoke these kinds of visual perceptions optically, and map out the circuitry. But I'll tell you the truth, the circuitry for me isn't like how one connects to the other and how they all connect together as a system, like I showed you at the beginning. I don't really care. I love what flies can see. I love figuring out what flies can see. I love challenging primate and motion vision scientists to say, hey, whatever your monkey can see, my fly can see. And I love to challenge fly vision theorists and say, what your model does not see what I know our flies can see, and they all get in a huff. And I love showing that there's object detectors in the fly brain that rival the hypercomplex cells in the feline cortex. And what are we going to do with all that? Hopefully someday understand why flies use it, but frankly, for now, that they exist, that flies make these computations, blows me away. So I'll leave you with that. These are the people that contributed to the work in STAR. So Jean Mongeau has his own lab. Mehmet Kelesh is graduating soon. He's going to go on to do a postdoc. Carol Estelle is a postdoc in the lab now. Jacob Aptekar bailed out of science. He's now consulting for McKinsey because the poor guy gets bored in 5 minutes. So every 5 minutes, you have to give him something new to do. So science isn't for him. Thank you for your attention and for staying. And we can talk about whatever you want if you have a minute. Thanks. The neurons and the pathways that do see that everyone says, okay, now this is a motion detecting element of the pathway. Actually, when you really push those, networks. They can respond and see things that the modeling does not predict. So, the short answer to your question is absolutely, a network-based approach that can predict where the limitations would be and the constraints would be more useful to me than the current neurobiology that we know, because we pose hypotheses of and right now, we're woefully shy of rigorous testable hypotheses that are built on neural computations, at least in the fly. Good idea. Hi.
So what's driving the interglomerular GABAergic cells?
What's driving the interglomerular GABAergic cells? So this 29G10 neuron, we think that it's really simple. So I think that neuron basically is an integrator, much like the wide field neuron that I showed in the first loop. Right? So I think that it turns out that when you label that neuron for synaptic markers and dendritic markers, they completely intersperse. So it's a fascinating cell. Basically, there's only 6 of them. Okay, so there's 6 of them. We don't know how big the dendritic field is for any one of them, but any one of those 6 GABAergic neurons is taking input from hundreds, or at least dozens of local columnar inputs. And the synaptic outputs and dendritic inputs are interspersed massively. I mean, there's no dedicated input domain and output domain. They're completely commingled. So I have no idea how it works, but I think fundamentally how it works is the columnar inputs come in that detect changes in light as it moves across the retina. And the neuron essentially computes the wide field. It's summing all the local inputs, and that's why it's responsive to movement of the whole panoramic. Right? It's integrating over all the local inputs. You get the largest response when you drive the most inputs. And so that seems very clear. How that gets transformed into a local domain-specific inhibition of LC11, individual LC11 neurons, I have no idea. It must be local in order to maintain the sort of end-stop inhibition. I don't know how it works. We don't know. The advantage that we have, though, in this system is that, so you saw 3 feature-detecting neurons. Fundamentally, they're exactly the same. They live in the same neuropil. They're columnar, so they're structurally similar, they're anatomically similar. They projected discretely, discrete glomeruli. So they live in the same processing domain, and yet they encode completely different features of the visual input. One is end-stopped, in that it's in size to small objects. One prefers elongated objects. One is agnostic to the two. So there's clearly an algorithm there that's being implemented by a cellular architecture. It's really pretty straightforward and simple. We should be able to have all the rules and all the molecules, and the molecular logic, the physiological logic, all worked out for at least these 3 cells. And I hope that it'll give us some general clues about how feature detection works in a sort of anatomically constrained system. Long answer, short question. Yes?
Last question. When you describe the fly visual processing as being sort of an order of magnitude faster than the neural, which I believe, but then you show the temporal kernels that are on the order of about 100 milliseconds, which is kind of slow, right? So it's an interesting puzzle there, right?
So there's 2 kernels. The slow kernel it persists for seconds. It's impossibly slow. Believe me, we got so much flack. Referees are like, this can't be right. You can't tell me the fly's got a perception in the last 2 seconds. No, that's not what we mean. But the fastest responses are on the order of 20 milliseconds. So if you measure a kernel of wide field displacement, it rises to half max in 15, 18 milliseconds. And this is the behavioral output. Right? This is how many you've got. You've got transduction of the visual stimulus. You've got 4 synapses minimally before you get out of the visual system, the optic neuron. You got to descend, project that neuron down into the spinal column, activate a motor neuron. Motor neurons have to activate the muscles, and all that's got to happen to move the wings, and there's just no time, right? So it's interesting, but it's very fast. And part of the reason it's fast is that the transduction kinetics in the retina are the fastest. So the retinal kinetics are faster in flies than any other animal measured. And that's where where the speed really comes from. The slow component, all that means is that if there's some feature off here to the right, the animal's sort of FM system sees that and slowly turns the animal toward it. And what that means is that the slow component of the response is superposed with the fast system. And so what you wind up with is that the slow kernel gives you a phase delay that you see in response to some of these complex stimuli. The simple stimuli, there's no phase delay. The animal just steers at it. The more complex stimuli where the bar moves one way, but the motion moves this way, one system is telling the animal to go left, one system's quickly, the other system is telling the animal to steer right slowly. When you add them together, then the animal turns right with a phase delay. And you can tweak the phase delay by up-down modulating the two curves. So it's real.
All right, we should end there. Let's thank Mark one more time.